All right. Good morning, everyone. Thank you for your patience. And I will be talking to you today about modeling Parkinson's disease, a long march to progress. On the next slide, I'm going to be introducing Parkinson's as a neurological disorder. And I should note, and I want to thank Julian for inviting me to this talk because this is National Parkinson's Awareness Month and it's an opportunity to kind of take stock and see where we are and where things are headed. There are a lot of neurologists in the room and clinicians who don't need any introduction to this, the study of Parkinson's, but nonetheless, it's necessary to go over some of the basics. So, for example, the pathological hallmarks of Parkinson's as we understand them, at least three of them. Two of them I've highlighted here. One is the formation of Lewy bodies, this dense inclusion that's the brain of patients. And then the loss of dopaminergic neurons of the substantial nigra pars compacta. Now, I should note that eventually you would lose a lot of neurons just grossly across the brain regions, but preferentially, at least at the initial stages, you've got nigra dopaminergic neurons being lost. And then it's necessary to also point to you that mitochondrial dysfunction and oxidative stress caused by free cytosolic dopamine are key characteristics of Parkinson's disease. There is no known cure. And I think for many people who suffer from the disease and their families, this remains really the point that people want to get to when you talk about the study of the disease. And on the next slide, um, I introduced to you the many, many facets of Parkinson's disease. And in fact, this is not exhaustive as a perusal of this list would show you. The resting tremors and postural instability are very well-known model symptoms of Parkinson's disease. However, there's a whole other world of symptoms that we call non-model symptoms that are just as important. In fact, in the past decade or two, they've emerged as a very key drivers of Parkinson's, so much so that many efforts to develop diagnostics have focused on this because they occur sometimes decades before the motor symptoms emerge. One of them is the gastrointestinal motility issues, right? And so there's this idea of a gut-brain axis, the connection between the gut and the brain. And many patients actually report that they have these issues many, many, many years before Parkinson's itself. As we know, it's really kind of becomes prevalent. Loss of sleep is another one. Anosmia, loss of sensation in the nose as well. So there's just a whole world of understanding that has come out of these non-motor symptoms. I'm going to be talking to you also about the environmental determinants, specifically rotenone, but there's a lot of compounds. There's a whole host of chemicals that we're exposed to in the environment. Thankfully, some of them not as much as we used to be, that raise your chances of getting Parkinson's disease. And finally, but not least among all of these facets, is this a kind of a relation back to the non-motor symptoms I mentioned earlier on, and is this idea that there is a seeding of this protein called alpha-synuclein that happens at points very distal to the brain, but through a prion-like mechanism finds its way up to the brain and then it kills with neurons as it travels along those paths. This is the basis of a new, relatively new model called the preformed fibril alpha-synuclein model that Virginia Lee's group at Penn kind of pioneered and has been leading the field in helping us understand how the disease unfolds. And on the next slide, I'll be introducing to you sort of where the model organism, my favorite model system, kind of fits into all of these. And for that, I want to kind of really introduce to you sort of like a retrospective of where we've been with Parkinson's and where we're going. So if I'm going to click on the next button, you will be able to see that there have been two milestones in terms of research and treatment. In the 60s, it's impossible for me to overemphasize this paper, Cortez et al., the New England Journal of Medicine, that really kind of has led the way to where we are right now. And in fact, when you hear L-DOPA, many people understand it as Cinemet, but it's this precursor to dopamine that is used to really treat Parkinson's disease. And the mechanism of action I'll kind of go over in a minute. But what I want to emphasize is that that point was where there was some kind of relief for patients that suffered from Parkinson's disease. And then about 30 years later, was the identification of the first quote unquote Parkinson's disease gene. Is this protein called alpha-synuclein, which I have mentioned before in a couple slides away. But this paper was also a very big deal. I have it on the slide, Polymeropoulos et al. Science paper. I forget now which patients that the paper really kind of looked at, but it was the first identification of a genetic, what we now understand as a causal factor for Parkinson's. 3 years later, Melfini's group at Harvard published the first fly model, and so it was a very clever trick that they pulled, which was to take advantage of this Human Genome Project, this wild time in our history in the sciences where the human genome was sequenced for the first time. And so the sequence of alpha-synuclein was essentially genetically engineered into the fly. And the question was, could you recapitulate symptoms, at least the motor symptoms of Parkinson's disease in Drosophila? The answer was a big yes. And surprisingly, that data has survived the test of time. It was controversial for a little bit, but I think that we've settled at a very nice equilibrium, and we understand a lot more about how alpha-synuclein leads eventually to Parkinson's. 4 years later, in 2004, the first environmental model in Drosophila was published. And these, again, learned from the mammalian folks, this time Tim and Terry's group, who were using rotenone to induce neurotoxicity in the rat model. And so on the next click of the button, I'll show you, and one more click, I'll show you that in 2024, I would love to be able to say that we've solved Parkinson's and things have moved on, and but that's actually not the case. There was a very big news, I think around 2013 or so, again from Virginia Lee's group, that looked like we might have had an antibody that could be used to successfully intervene, but that kind of didn't work out as everybody had hoped. But what it does tell us is that there is a way forward, right? We know a lot. We have a lot of tools that we couldn't even have dreamed of in the 60s that really kind of points us the way forward, that we can find not only ways to better test the drug, but to not only ways to better identify or detect Parkinson's early on, but to actually find an intervention. And on that note, I will spend the next couple of minutes or so talking about my own work and how it fits into the broader field. And so this slide really kind of gives you just a very generic introduction to the dopaminergic system in the context of a single synaptic vesicle release site. One of the problems with L-DOPA is that if you click on the button, you will see that, and another click, dopamine is a great drug, right? It does wonderful things, but it really also kind of like causes neurotoxicity in those neurons. So it can kill the neurons while trying to potentiate them. And so we think that we found a way around this, and it's that the two red balls with the it's a small machine called the vesicular monoamine transporter. It's a protein whose job is to package dopamine. And so one thing you can think of is you want to release dopamine, but another way to think of it is you want to get rid of dopamine from the cytosolic space so that it doesn't toxify everything. And so we use rotenone, which I talked to you about earlier on, that was first identified in Tim Greenmeyer's work and Terry Hastings as well. So we use that in our lab to kind of induce a sort of an oxidative stress state. And we ask the question of whether we can lower that stress state by increasing the efficiency of that protein, vesicular monoamine transporter. And so in a few clicks now, up to the next slide, and we tested that model. So this is my data from when I was at UCLA. I was doing the Drosophila side, but the mammalian folks were doing theirs. Zebrafish people were doing stuff. And one of the convergence was that depending on, regardless of the system, you could actually use this vesicular monoamine transporter protein to save these neurons from neurotoxic damage. And then in the next slide, I show you some of the very interesting data that has come out from our lab and also that of our collaborators at UCLA. So we've been doing the Drosophila side of the work, and we show that you can actually, there's a particular compound that we think it acts by chemically increasing the activity of that protein, vesicular monoamine transporter. And so we are able to rescue to a small measure the toxicity that would normally be induced by rotenone. And then the mouse people at UCLA have actually gone a couple steps further, and they've been able to test Parkinson's disease models in the primary dopaminergic neuron cultures. They use alpha-synuclein, that gene I told you earlier on, to induce neurotoxicity. And then in a separate experiment, they used the environmental stressors. And in both cases, they found that dacarbazine worked really great. All right, so in the next slide, I'll kind of give you a little bit of a downer, which is like, okay, this is all great, so why are you so discouraged? The problem is that the carbazine is also very toxic. It's a chemotherapeutic agent, so you don't want to choose between cancer and Parkinson's, right? It's not a good choice to have. So what we've been doing in this slide is that we've actually looked for a circumvention of the carbazine itself by looking for this compound called ACRE. It's a structural isomer, structural analog of the carbazine. It doesn't have the toxic moiety that is used to fight cancer with the carbazine, but it does, we think, have the dopaminergic potentiating aspect of it, which is the most exciting part of this. In fact, on the slide on the right, the data on the right is collaboration with Gary Miller's lab at Columbia University. We met last summer at a conference in Switzerland, and this collaboration started. And the data here is the first time actually we've been able to show that you can use this compound ACRE to increase dopamine release. So this is very great news, because if it works out as we're thinking, then we have better way to treat Parkinson's than just L-DOPA. That's on the low hanging fruit side of it. But further on, we can actually look for ways to actually develop disease modifying therapy based on this knowledge of sequestration of dopamine and its efficient release. And so I'll go on to the next slide as I wrap up to just summarize that there are many, many aspects of Parkinson's on this National Parkinson's Disease Month. There's still a long road to go, but there are many, many strategies out there that we think can actually lead to better treatment. So what I've just showed you is one part of it. There are others that are working on some interaction between Parkinson's and Gaucher's disease, using therapeutics that may be effective in that disease pathway to intervene in Parkinson's as well. And so I will stop there. My next slide is just to acknowledge my team and all the people who helped me, and I'll take your questions.
Thanks, Hakeem. It's wonderful. And thanks, Jackie,